Hi, everyone. This is Nasha Fitter from the FOXG1 Research Foundation, and we have kicked off a new series where we explain all of the avenues of science that we are tackling to find a cure for FOXG1 syndrome. Today, I'm going to talk about FOXG1 repurposed small molecule drug screening, a lot of buzzwords there that I'll go through and talk about the projects that we're doing within this category. So first and foremost, what is a small molecule? A small molecule, very simply, is a drug that can enter your cells easily because it has a low molecular weight. And small molecules that we take every day are examples like Advil and Tylenol, really easy to get into our system. Now, the beauty of small molecules is that there are thousands of databases available of various small molecules that we can test on our different assets to see, could they help FOXG1 syndrome? What we want to do is test databases of repurposed drugs. That's another buzzword. You hear it all the time. Do you have a repurposed drug? Are you testing a repurposed drug? So something to keep in mind is, small molecules, drugs like Advil, Tylenol, etc., they could take over 10 years to get to the market. There's a lot of regulations and safety, etc., that are important before we put any of these drugs in our children. Repurposed drugs have already been tested in people, usually, and they have definitely passed safety tests. They tend to be drugs that are either off patent, they've been approved for another disease, but they could work for FOXG1 syndrome, or they were kind of stopped at a point by a company or a researcher. So different academic institutions, different companies have massive libraries of repurposed drugs that are available, and we could use them to test. Now, if we find one of these drugs that works on any of our symptoms, seizures, movement disorders, speech, anything, we can very quickly get it into a trial. And that's really the beauty of a repurposed drug and why we're excited about it. Now, how do we screen these repurposed drugs? And screening is another buzzword you hear all the time. So there's two main types of screening. There is high throughput, and that's where you could take thousands or hundreds of thousands of drugs even and screen them. And one of the platforms for screening that we're using for FOXG1 is zebrafish. So this is a project we have underway in London at King's College with Dr. Corinne Huart. And zebrafish are kind of amazing because they're inexpensive, they're very, very small, and they're transparent. So you can actually see how the drug is going through different organs. It gives you a good picture right away. And you could screen thousands and thousands of them very quickly. The second is what we called a neuronal screen. And many of you have donated your children's skin cells to Coriell, and this is why it's so important, because we take those skin cells and we turn them into something called an induced pluripotent stem cell. And these stem cells then can be taken apart and we can do different testing. So one of the things we do is we actually can differentiate a neuron from that stem cell and do high throughput testing on those neurons to see, can these drugs affect our children at a cellular level, at a molecular level? That's getting a little deep into the science, but hopefully that makes sense. And then there's low throughput screening. So let's say you screen 2,000 drugs and you find, oh great, 10 of them seem to have some prospect. Then what we would do is screen them in our mouse models. And most of you know, we have a whole host of mouse models that are being developed with Dr. Sue Kyung Lee and Dr. Jay Lee at our FOXG1 Center of Excellence at the University of Buffalo. So we would then test those drugs there to see, can it relieve any actual symptoms in these mouse models? We also have 3D brain organoids that we have created at the University of California, San Diego, and we can also do testing on those. The reason we cannot do a high throughput screen on a mouse model or a 3D brain organoid is these are more complex assets. They're more complex models. It takes a long time to screen, it's more expensive, and you get much richer data, which is what you want when we get to the point of, hey, could this actually work on our children? So those are the assets that we're going to be screening on. Now, what are the different types of screens we're doing? So the two basic ones, and this is what most groups do, is, can we affect the protein? So FOXG1, all of our children have a mutation at the genetic level, but what it actually does is it affects the protein, the FOXG1 protein, and that's what tells our body what to do. So can we increase the level of FOXG1 protein? our children mainly have a decrease in FOXG1 protein. Can we also regulate something called IE imbalance? So IE, many of you will have heard the term GABA, glutamate. This is what that means. So a mutation in the FOXG1 gene has severe impact in our GABA glutamate balance for our children. So can we find drugs that could regulate that? And that could help many symptoms. We are just embarking on a new, what is really a groundbreaking screen, because it truly hasn't been done in science. Just experiments have been done, but we think this could work for FOXG1. So what makes FOXG1 special is it is a transcription factor. Some of you may have heard of that. What does that mean? Transcription basically means to transcribe. So FOXG1 tells other genes what to do, when to turn on, when to turn off. And because of that, even though FOXG1 is such a teeny tiny gene on one chromosome, it can have such a massive impact in our body because it tells hundreds of other genes what to do. So what we want to try to understand is, what is FOXG1 transcribing in these other genes? And we will look at things like RNA expression levels. How much RNA is it transcribing? We can then measure that transcription level and then do a neuronal screen to see, hey, if we administer a whole host of drugs, could it fix that transcription level? And then again, if it works, we would go and screen in our mouse models, in our 3D organoids, to understand, does it relieve any symptoms? So that's a new groundbreaking screen, as well as these other screens we're doing. And our goal is to try to find something as soon as possible to fix as many symptoms as possible for our children. You can follow our progress on our FOXG1 Research Projects page, which is on our website, as well as there's a ton of other information on our website. And we will be doing more of these little bite-sized science pieces moving forward. If you have any questions, of course, please reach out to us. All our email addresses are on the website. Thank you very much.